This is a brief introduction to karyotype analysis, which is the second half of our genetic counseling notes in Biomed. And so I just want to run you through a couple things about what a karyotype is, and then we'll take time to really do a task using karyotypes, just to kind of give you an idea of what that is like in a clinical setting. So we've already discussed in this unit a couple different things. A human cell has 46 chromosomes normally, and those are in pairs, right? Because you get 23 chromosomes from your mom, 23 chromosomes from your dad, and those pair together. Now, this would include 22 numbered pairs of chromosomes that we called autosomes, and will also include a single set of sex chromosomes. And this is what, if we could get a really good electron microscope image of the chromosomes when they are condensed and duplicated and ready for cell division, this is what they would look like. And so you can see sort of this X-type structure that we oftentimes draw in class when we're looking at chromosomes and cell division. The question here is which ones pair with which ones? Now, before we get to the answer to that question, we have to kind of ask the question of how do we visualize chromosomes, or how do scientists do this? Now, the first thing we have to realize is that chromosomes have to, it's a requirement for them to be in their condensed form. So what they would look like kind of at the start of cell division in order for us to see them. If they're not in this condensed form, so like during interphase, for example, the chromosomes aren't really separate chromosomes. They're actually just these bundles of genetic material that we call chromatin, and we can't really tell one from the other. But if we take particularly a white blood cell, which is what these are, the kind of cool looking cells, these are 5 different types of white blood cells here. If we take these white blood cells, we could find them under the microscope. They're very commonly already in cell division, and then they're also really easy for us to culture or sort of grow copies of in a laboratory setting. And once these cells are in mitosis, and particularly when they're in the last part of prophase or maybe they're in metaphase, we can sort of chemically freeze the cell in that stage, and that allows us to extract out those chromosomes and actually study them. Now, once we've extracted the chromosomes, we can go back to my initial question, which was, how do we tell which ones actually pair together? And there are really 3 ways that we can do this. The first way is to look at the length or the size of the chromosome. Homologous chromosomes are essentially the same length. And the 22 numbered autosomes are actually numbered based on their length, with chromosome 1 being the longest and chromosome 22 being the shortest, give or take. And when we look at these in a second, you'll see that some of the long ones are really close in size and some of the shorter ones are really close in size. But technically, we number them from 1 to 22 from longest to shortest. Now, the second thing that we can look at is centromere position. So if you look at this picture over here, the centromere is sort of like an indented region. It's usually fairly central, but it's never right in the middle. And if you recall, if that word sounds familiar, centromere is the area on a chromosome where it can attach to the spindle to move around in mitosis or meiosis. And so that centromere will be at some position along the chromosome, and it is typically in the same position if you're looking at 2 homologous chromosomes. Now, wherever that falls, you're going to end up with kind of 2, we say there's 2 arms on the chromosome. The shorter arm on one side of the centromere is called the P arm. The longer arm is called the Q arm. So technically, what we're looking at here when we say centromere position is we're also looking at the length of P versus Q, which again should be similar if the chromosomes are homologous. The third and final way and sometimes kind of almost the best way to identify which chromosomes match with which is called banding pattern. Now, what you'll also do when you extract chromosomes is you will stain them with a stain, kind of like we stain things that go under a microscope. And when you stain them, the stain will bind to some regions of the chromosome more than another. But what's cool is that if the chromosomes are homologous, they will stain or band in almost exactly the same way. So the striping of the chromosome should also be almost identical if you're looking at two chromosomes that are homologous. So for instance, chromosome 1 and the other chromosome 1 should have pretty much the same banding pattern. And I put this picture of a snake not to creep you guys out, but that's also a way that we can, similar analogy here, right? Another way that we can identify snakes is by how they are striped. And so when we look at chromosomes, if they have the same kind of stripes, they are probably a pair of chromosomes. So, what is this whole karyotype thing? A karyotype is really just a picture, right? A karyotype is a picture of all of the chromosomes from a cell arranged in their pairs so that you have a nice little organized view of the different chromosomes. Now, traditionally, this was done by getting a cell into mitosis, taking a picture, literally through a microscope, of those chromosomes, then actually printing out a blown up copy of the picture and literally cutting out the chromosomes and trying to pair them up based on their size and their banding pattern and where their centromere was. Now, today, as you might guess, our technology is better and we can do this stuff with computers, right? And so now we can get a picture and we can digitally rearrange the chromosomes, and the computer can help us pick out which pairs, which chromosomes go with which chromosomes. But in the end, you're going to get pictures that look like this. So here are a couple of examples of karyotypes. So this would be, one cell over here, and this would be a different cell over here. So what this allows you to do is quickly look at the sort of a spread of the chromosomes and their pairs. One thing you realize right away is this is an actual picture of the chromosomes. So they're not going to be perfectly nice and straight and lined up for you, right? So for instance, like this right here, there's nothing wrong with this chromosome. It just happens to be a little bit bent in the picture. Same with any of these that are kind of bent up in different ways here on this picture. So that makes it a little more difficult to figure out which one's which, because it's a little hard to see their length. Remember too, we look at centromere position. In cell number 2 here, it's actually a nice karyotype where it even shows you where the centromere is with this little like dashed line for each one. Otherwise, you just have to look really closely and you look for little indented spots, kind of like right in this area here on cell number 1. That third thing is banding pattern, right, or the stripes. And if you look at some, it's more clear than others. But check out like these two here, this chromosome 7 and cell number 1. You see a good stripe here at the top, really solid one here, much lighter one there. And so you can tell that those patterns actually match up together. Now, the question that we often ask is how do we classify these things? Now, to do a classification, you do the total number of chromosomes followed by what we would call the sex complement. So, for instance, this one on the left here, cell number 1, this would be a normal female. And the way we know that is if we skim this whole thing, we're going to see a total of 46 chromosomes, and we're going to notice that it has 2 X chromosomes. So we would classify this one as 46XX, which would be a normal female. Now, if we look at cell number 2, right, notice again, if you scan it, that you've got 46 total chromosomes. And then check out their sex complement, XY. So this one would be a normal male, and the way we would classify that is 46XY. All right, so these are what the normal ones would look like. Now I'm going to get rid of some of my writing here, and we're going to have a couple other possibilities. I'm going to draw a little extra on these and see if we can figure out what's what. So for the first one, let's say you look at this karyotype and you notice that you've got something like this going on. Now, when you look at this karyotype, you notice, hey, wait a second. There's 3 copies of chromosome 21. So now what we're looking at is a different classification, right? This one is going to have 47 total chromosomes, but it's still XX for a female, right? And so we could classify this as 47XX, and we give a little more detail. That's trisomy 21 or Down syndrome. So let's do another one. Let's say that you look at a karyotype and you see something like this. All right, so you look at this one and you say, wait a second. It's got 3 different sex chromosomes. So for this one, again, we've got 47 total chromosomes because we got an extra one. And this one's going to have a different sex complement, right? This one is XXY. Now, believe it or not, this is still male because it has a Y chromosome. But it's got 2 X's and a Y, and so this is going to be a different disorder called Klinefelter syndrome. Now, with this, you're going to move into a task. On that task, you're going to use some of the information from this along with the internet, where you can search and try to find more information to try to categorize and identify some other disorders. Good luck.